So good afternoon, everybody. Thank you all for being here. May I start by reminding you, for those of you who do not know where you are, that this is the session entitled, "It was a joke." From guidelines to practice, proven strategies for implementing quantitative train of four monitoring. The session is supposed to be 1 hour long, but we'll be here for 3 extra days to respond to any of your questions. I'm sure there will be many. At the end of the presentation, we will have a Q&A, question and answers session. And you can approach the microphone over there and pose your questions. Hope you won't stump any of us. We have the pleasure of introducing 3 panelists today. One who is representing the academic anesthesia community, Dr. Carlos Artime. Someone who's representing the community practice, Dr. Aaron Persinger, and a third panelist who's representing the pediatric anesthesia, Dr. Yuri Solnoki. So just a couple of reminders, if you don't mind. The description of the symposium is to discover strategies for implementing quantitative train of four monitoring. The 3 settings I've already alluded to, but I think that they're applicable really to any other setting, whether it's outpatient or NORA or really in any patient who's receiving neuromuscular blocking agents. I would also ask to please silence your iPhones. It's never good to have them go off in the middle of a talk. And also feel free to shout bravo at the end of every presentation. I think they will deserve them. The breakdown of the session is to start with Dr. Charlie Artime. But before we go there, I just wanted to have an idea of how many of you actually use quantitative or objective neuromuscular monitoring. Wow. For those of you who raised your hands, you're excused. So it's a pleasure to introduce Dr. Charlie Artime, who is Professor of Anesthesiology and Executive Vice Chair of the Department of Anesthesiology, Critical Care, and Pain Medicine at the McGovern Medical School at UT Health, Houston. That's in Texas. His department uses quantitative neuromuscular monitoring across more than 100 anesthetizing locations at Memorial Hermann Hospital, Texas Medical Center, one of the busiest level 1 trauma center in the US. He leads a department of over 300 physicians, trainees, and advanced practice providers. And he brings a unique perspective on the opportunities and challenges of implementing new technology within large, complex academic systems. The second speaker is Dr. Aaron Persinger, who is a private practice anesthesiologist and partner at Boulder Valley Anesthesiology in Boulder, Colorado, where he practices across multiple community hospitals and surgery centers. He serves as the medical director for both the Department of Anesthesiology and the pre-anesthesia testing clinic at UC Health Broomfield. Dr. Persinger has led the perioperative implementation of several new anesthesia technologies, most recently introducing Senzyme's Tetragraph across 3 hospitals. At Boulder Community Hospital, one of the first facilities in the US to fully adopt the next-gen Tetragraph. And quantitative monitoring usage has improved patient safety and reduced Sugammadex pharmacy spending by more than $100,000 annually. And finally, Dr. Judith Solnoky, who's a pediatric anesthesiologist and chair of the Department of Anesthesiology at Nemours Children's Hospital in Orlando, Florida. She has led the implementation of quantitative neuromuscular monitoring across all operating rooms at Nemours, driving improvements in patient safety and standardization of care. A native of Hungary, she received her medical degree from Semmelweis University in Budapest, completed her anesthesiology residence in Syracuse, New York, and a pediatric anesthesiology fellowship at Children's Hospital, Colorado. She holds a master's degree in patient safety leadership and a coaching certificate from Erickson Coaching International. And she really is passionate about servant leadership, mentorship, and developing young faculty. So let's start with the first discussion by posing a question. Why is it this late in our practice? Why should we change what we currently do if we're not using quantitative monitoring? So I'm going to ask the first panelist to take that. Judy, do you want to start?
Sure. You guys can hear me? I think I approached this with the hospital from a patient safety standpoint, and I'm lucky that I practice in an environment where patient safety is paramount, and certainly having the ASA statement, having the evidence that how this is superior to what we used to practice was very helpful in the discussion. I also have to add that in the past decade or so, as the concern with neurotoxicity in the pediatric population came up, I believe that we started to use muscle relaxants. So we would use truly a combined technique for the longest time. And in my training, we pretty much used the sevoflurane for muscle relaxation as well. And that has changed in the past decade. And with that, now we use more muscle relaxant, and we were not good at monitoring it. And I believe that this technology allows us to do a more precise anesthetic. And again, that discussion then led to our hospital approve us to trial some of the technology, and then we implemented it. We're certainly not fully done with the implementation because we're still trying to integrate it with our electronic medical record. However, I think we delayed with that because we didn't think of it ahead of time.
Thank you. Charlie, do you have stuff to, I'm sure you'll have things to add?
Yeah. So, for us, it's really been a long time coming to implement quantitative neurophysiologic neuromuscular blockade monitoring in our institution. We've known for a really long time that as anesthesia clinicians, we don't really do a great job of dosing paralytics and reversal. Plenty of studies out there for the past 15, 20 years showing the risk of postoperative pulmonary complications with inadequate reversal and the prevalence of postoperative residual paralysis. Even with Sagamidex, this issue of postoperative residual paralysis hasn't gone away. We had in our own institution a pilot QI project using quantitative technology that showed even with 90% Sagamidex usage, we had as high as 15% residual paralysis rates in PACU that was irrespective of the dosing of the reversal agent chosen. And there's been other studies in the literature recently to kind of support that across other institutions. So it's really been a long time coming, but I think it was two things that really has driven, why now, why quantitative monitoring. The first thing is the availability of this technology, right? This EMG-based quantitative monitoring technology really allows us to monitor twitches in patients during surgery practically, right? Whether we have access to the arms or not, arms can be tucked. old acceleromegraphy technology really wasn't conducive to that. So the practicality of EMG-based technology has really facilitated. And then the second thing, as Dr. Solnicki mentioned, was the ASA practice guidelines. Now that we have those guidelines, it's really given us the ammo to go to our hospital partners and say, hey, these are the new standards of care. These are the new practice guidelines. We were able to get quantitative monitoring now.
Thank you. Aaron?
Yeah, so there are several reasons why I think several of us think now is a great time to implement. I'll try to just quickly highlight maybe my top 3 or 4. I always have several, but we noticed the same thing at Boulder that Dr. Artime was talking about, which is even if you're a high Sugammadex using facility, you have patients in PACU, your rate of residual neuromuscular block, may still be 15%. And so we started to ask ourselves if that was really good enough for us to have 15% of the patients be weak, because if they're in that position, they're at risk for respiratory complications. And so what do we know about weak patients and respiratory complications? I think 2 great studies to look at. There's a lot of them, but there was a study done at Temple. I think Lori Edwards was the investigator on that one. And they determined how often respiratory complications would happen, which was like 2% of all cases in that study, and determined and how much they cost, which is if you get a respiratory complication, it adds $30,000 at least to that episode of care. And then Wade Weigel's group at Virginia Mason showed that if you institute quantitative neuromuscular monitoring, you can reduce those pulmonary complications by 43%. And not just that, but you can lower the PACU stay by 7%. And you can also lower the hospital length of stay. And so we thought at our facility that was something that we wanted to go after and try to capture, was numbers like that, reduction in pulmonary complications and shorter PACU stays and increased safety for the patient. So before we started it, I tried to take a look at where are we at with monitoring before we did the implementation. And you have those old peripheral nerve stimulator boxes from 1970 that people maybe use or not use. And unfortunately, the statistics on those is that 40% of anesthesia clinicians actually don't even use that gray box. They actually don't use any monitoring at all. They just give the reversal agent and rely on the fact that Sugammadex is so good. But if you think about it, we don't do anything else in anesthesia that way. You don't give a medicine to treat hypotension and then not check another blood pressure. That would be a silly thing to do. So I've never understood why it was okay to do that with neuromuscular blockade. And so I thought the reason that a lot of people weren't using the gray box was that maybe it wasn't a very good tool. Maybe it wasn't giving them the information that they needed, that was actionable, that was helpful, that they can act on. And so when I saw some of the quantitative monitors, the newest iterations, and how good they were, it was kind of an aha moment for me. I'm like, okay, yes, I think people can use this because it's fast enough, it's reliable enough, it's a workflow that they know. So I knew that my team knew how to put a BIS or a SEDline sticker on the forehead to monitor the brain. And so I knew that in their workflow, it would not be too hard to get them to do the same thing on the wrist with the neuromuscular junction. And then the last thing was that we thought to ourselves was, now that the ASA guidelines are out and they strongly recommend quantitative neuromuscular monitoring, if we had a partner, a doc, or a team member come to us and say that they wanted to follow the guidelines, but we didn't have the tool to give them, we felt like that was unacceptable at our facility. We're like, we have to at least have the tools if they want to practice evidence-based medicine and follow the guideline.
Thank you very much. Outstanding. I think it's pretty unanimous that regardless of setting, it's really important that for those of us who did not use maybe quantitative monitoring, why would we would want to change? So now that we've established that, maybe we can go to each one of the 3 experts and ask them how they implemented quantitative monitoring in their settings and their institutions. And maybe you, Dr. Artime, can start.
Yeah, so as Dr. Brill mentioned during the intro, we're a large academic medical center, 100 anesthetizing locations. And, considering the number of anesthetizing locations and kind of the wide geographic footprint, we really felt that the best way to implement quantitative monitoring was to have one of these devices dedicated to each anesthetizing location. So we have one device attached to a pole or attached to the anesthesia machine in every anesthetizing location. And several things are important to really have a good implementation and a good adoption with the team. The first thing is education. You know, a lot of our clinicians know the risks associated with postoperative residual paralysis. They know about the ASA guidelines that came out a couple of years ago, and they know the importance of this. What they maybe don't know is how to practically use the technology, how to practically use the technology to drive decision-making with respect to dosing of paralytic and dosing of reversal agent. So a big educational push is really important when you're implementing this. You know, grand rounds, presentations, emails, et cetera. The other thing is protocolization. So we developed protocols to kind of guide reversal using quantitative monitoring. You know, leveraging the ability of seeing an adequate recovery to decrease Sagaminix usage, and we can talk about that a little bit later. But protocolization really helped, I think, our providers implement this technology. And then lastly is just support from the device team. So, you know, when you're rolling this out, we had great help from the device, you know, company coming out, making sure our providers knew how to use the device, helping to troubleshoot, teaching our anesthesia techs how to help troubleshoot, and that's been really important.
Thank you. Dr. Persinger?
I had the opportunity to implement at 3 different hospitals, and then I was able to see or be aware of some other implementations at some other sites that didn't go as well. But at the 3 places where I was able to help out with an implementation, it kind of went just a little bit different way at each facility, which I thought was kind of helpful because, you know, there may be different ways that it goes for different people. So one of the first hospitals that I implemented at Boulder Community, they took the all-in approach, kind of like Dr. Artime mentioned, where they decided that putting one in every anesthetizing location was the best thing to do. They thought about that for a while, but after looking at the economics and use case and safety benefits, they're like, okay, let's go all in. And so they implemented everywhere, which is definitely my favorite preferred approach, because that education piece that is so critically important to spend time helping everyone learn how to use the device and become very facile with it. It's much easier if it all starts at one time in all the locations, and then you don't have to have devices traveling from one location to another where you could lose a cord or damage a cord or something like that. But I had some other facilities, and this was interesting, where for some places, depending on how much money is being invested, that might become like a capital expense for them, where they have to fill out certain paperwork, involve certain stakeholders, or get an approval. And sometimes they only do that at certain times of the year. And so if that's the case, like you find that you want to get going, but you're being told it's going to be some time before you can make a capital request, or maybe they're telling you it's not likely to make their top 3 to 5 issues for some reason, you can also implement 1 or 2 monitors at a time. So I did that for some smaller locations who weren't really convinced they had the ability to go all in all at once. And so, you know, just getting 1 or 2 at a time, sometimes that's below that threshold where you have to fill out all that paperwork. And so in the meantime, while you're waiting to get enough monitors for as many or all of the anesthetizing locations as you can perhaps have them living in your anesthesia workroom and have the techs get trained and become really good at the devices where they can bring them to you upon request. Yes, the only downside there is, again, the more those devices move, the more there could be some risk of some human factor damage, or somebody drops it or for whatever reason. So it's much better to have them mounted on your machine. And then you may have some users and non-users, and some patients are getting this great guideline care, guided care, and other patients aren't. And so those are kind of some of the many reasons why 1 or 2 at a time might have some disadvantages, but it may be better than not getting started at all, in case you can at least apply it to a few high-risk patients while you're trying to get all your monitors on board. But certainly, whichever way your implementation goes, I really became a fan of, if you can, engaging your biomed team to try to mount the device on the anesthesia machine itself, if you can, because it's a popular choice also to put them on IV poles, but you may have competing space on that same IV pole with like an Alaris pump or various kinds of syringe pumps that you move up and down, and then all of a sudden the different pieces are kind of banging into each other. And again, you might have inadvertent damage to a device. So I like to mount them on the anesthesia machine. And the other thing that I really grew to like by mounting them on the anesthesia machine, kind of somewhere like maybe it's off to the left of where your vital signs screen is on another pole that comes down, is then you can coil the cable that you hook up to the sensor right up there next to your pulse ox, blood pressure, and EKG cable, on like a fourth hook there. And then it serves as a reminder like, oh yeah, I'll place the sticker, I'll place the electrode. Or psychologically, it reminds people, oh yeah, actually monitoring my neuromuscular blockade might be perhaps as important as monitoring these other vital signs too. It's just kind of convenient. And then when you do your scan of the vital signs every few minutes or however often you do that, you can then pivot your eyes one screen over and check in a hands-free fashion what the depth of your neuromuscular blockade is, which can help you optimize your conditions for your surgeons and make sure the patients are safe.
Thank you very much, Dr. Zolnicki.
So we went with the route of implementing it every operating room and mounting it on the anesthesia machine. However, during the trial, we had it on the IV pole, and when we put it on the machine, I had several people who told me after that I didn't notice it. I didn't know it's there. So that took time for people to recognize that, okay, it's right here. And some old habits die hard, right? You have to pry out the little twitch box out of people's hand, which we did. Initially, we had both devices, the quantitative and the old-fashioned one, present. And I would walk into an operating room and see people putting the taser on the head. And I would ask, why are you doing that? And they would look at me that I'm monitoring muscle relaxation. I'm like, why don't you use the better technology? And they say that, the patient was already covered. I'm like, but when do you put your monitors on? Your normal monitors? Well, at the beginning of the case. Well, why can't you put this one on? Right? So a lot of sort of getting the flow down of how and when to apply the sensor certainly played in the implementation. And then finally, I was taking away their little old boxes and told the anesthesia techs that, okay, you don't have to throw it out just yet, but I want a list. If somebody wants it, I want to have them sign it out. Meaning I need to know their names. I need to know who needs further education in the newer technology.
Okay, thank you. Fascinating. I'm sure there will be a lot of comments and questions from the audience on this. Thank you very much. So now I think the meat of this meeting is really about implementation, the barriers, how you overcame them. I look forward to a lot of the audience participation in the Q&A session. So can we start, please? Let's start with you.
About the challenges?
Yeah.
Sure. As I mentioned, old habits die hard, right? So that was the first challenge to really integrate it into your routine of thinking about the monitor, of applying the monitor. And I have to say that the best success we actually had with our residents. The residents who came and rotated with us, and initially, of course, we show them that we have this technology, this is how you use it, this is what the numbers mean, and then the next time they would have it out, which means that the next attending working with them, or the next day an attending working with them, now that is going to be part of the residents' preparation, and they would use it. So we found that the residents actually really helped with establishing that culture, that this is just one of the extra monitors. They prepare everything, right? I have to admit that as an attending, as an experienced person, I prepare the minimum, and then everything else I can fix after. But the residents, they prepare everything. And having that sort of prepared for everything, and I have the monitor out or I have the sensor out, certainly helped. And with our again, physicians or APPs, it was showing them how useful this is and showing them how we didn't know our drugs as we were using it, how we knew a little bit of rocuronium in the setting of the pediatrics, of how this one dose that I'm thinking this is going to be just fine, maybe lasts forever in my patient. And how even reversal. I think I'm giving a dose that might or might not be adequate. Or I'm thinking that, oh, I absolutely need to reverse after X amount of time, and I see that my patient has recovered. So we knew little about our drugs and how we use it, also how long it takes for rocuronium in certain doses to kick in. We give it, and then we want to intubate, and it's maybe not time. So actually showing the benefit of having this monitor certainly helped with that change in practice.
Yeah, I'd love to hear some of the comments that and I'll ask both of you to comment on that as well. But given that the settings are somewhat different, well, they are different. What are some of the comments that are most memorable to you from those upon whom you're trying to?
So one day I walked into the operating room, and CRNA was telling me that, well, I don't think your monitor, right, not our monitor, but your monitor is not working, and she already put the taser on the head. And look, I have something here, and your monitor says 0, and I'm like, I don't know. I don't know if we should believe yours versus mine, if now we're breaking it down, your thing and my thing. I said, well, when we reverse, we'll see if my monitor actually works or not. And sure enough, when we reversed, the twitches came back. It was 0. It was 0. It was not a question if it was 0. She just she didn't believe it. It was in a child who had one single dose of a paralytic. And this is how little we know how it works in different age groups, that we assume that at this time I should already have a twitch back. Well, we didn't.
Yeah. Thank you. Dr. Artime, any barriers and how you have overcome them? I'm sure that your implementation was smooth as silk.
Yeah. So, you can imagine at a really large department, like I said, we have over 300 clinicians. There were some barriers, and there were some, and still are some issues with adoption, right? I think anytime you introduce a change to a practice, you have the early adopters, people who, see a new technology and they get excited about it and they want to start using it right away. And then you have the slower adopters, right? Who maybe need a little bit more convincing, takes a little bit more time. So, the strategies there are, one, continuing the education, continuing the contextualizing. Why are we introducing this technology? Why are we encouraging everyone to use this technology? It's about increasing patient safety. It's about following our National Society's guidelines. I like to use the example to our clinicians. Everybody in our department is really serious about following the practice guidelines for perioperative fasting and MPO guidelines. Well, the practice guidelines for the use and reversal of neuromuscular blockade from the ASA has the same kind of degree of weight as a practice guideline, right? And so, these are things that we have to kind of continuously remind people. The other barrier that we have, particularly with some people who are maybe a little bit more reluctant to start using a technology, is again providing context when perhaps the monitor for a given case doesn't work, right? Maybe the probes wasn't placed properly. Maybe there's some electromagnetic interference that's kind of preventing the use of the device. Perhaps the patient has some muscle atrophy or some denervation, and you're not getting a very good signal. And some people will see that and say, you see, it doesn't work, and they may not want to use it again. And I'll use the example is, everyone here has put EKG leads on a patient, and you see a kind of a small low voltage QRS, and the machine says asystole. Well, it's not asystole. That doesn't mean that you take your EKGs off every patient that you take care of for the rest of the day because you had one EKG that wasn't showing what you knew to be the case. So like all monitors that we use, it's just one data point that we have to take into consideration of the whole picture. And yeah, there's going to be times where any device isn't going to give you the data that you want. That doesn't mean you toss out all of the the entire technology, right? It's going to add value to your care of the patients in the majority of cases. And then the other thing is, another barrier obviously here is cost, right? Some of you might be in clinical situations where your department is burdening the cost of implementing this kind of technology. In our situation, we have hospital partners. We're an affiliated medical school with a separate hospital, so they're the ones shouldering the cost. How do you convince them that this is a worthwhile investment? And that's really where we've used quantitative monitoring to introduce protocols that allow us to use lower doses of Sagamedex in order to save money, right? When you look at the package insert dosing for Sagamedex, there's the emergency reversal for airway. We won't go there right now, but otherwise there's just 2 doses. There's a PTC of 2 dose and there's a train of 4 count of 2 dose. Well, there's a whole spectrum of paralysis and recovery that's not covered by those 2 points. And there's a lot of, there's a lot of real estate there. There's also the fact that the package insert dosing for Sagamedex is not designed to be the perfect amount of Sagamedex to encapsulate how much free rocuronium there is. It's extra Sagamedex to get really quick reversal. And there's many situations where you can give a lower dose of Sagamedex, wait an extra minute or 2, and get adequate recovery. So using quantitative monitoring and using protocols to encourage kind of in-between doses and some low doses, encourage dose rounding, giving a dose, seeing a response, and then only redosing if you're not getting adequate recovery has really given us the potential, particularly with aliquoting doses, which we've just started doing that with 100 milligram syringes of Sagamedex to decrease overall Sagamedex costs.
Fantastic. Yep. I'm into it. I believe you. Dr. Persinger?
Yeah. Anyone who goes to implement this, you'll find some barriers. And when I'm trying to think about how to overcome some of these barriers, sometimes it depends on who I might be talking to at the time in the perioperative care arena. So I find that sometimes people are speaking different languages depending on what group they're in. So I ask myself, are you speaking the language of patient safety and outcomes, or are you speaking the language of economics, or are you speaking the language of speed and convenience? And there's some overlap in who's in what group, and sometimes people are in multiple groups. But in general, for the language of patient safety and outcomes, a lot of times it's the anesthesia clinicians themselves where that's clearly their top priority. And your anesthesia team, like everybody, wants the same thing. They want a great outcome and they want the patient to do well. And so, but some of the people on the team also, they might be aware that the guidelines have come out, for example, but maybe they're not aware that that when you actually read those guidelines, that it recommends quantitative monitoring of your neuromuscular blockade, and the level of evidence is very high, and the strength of the recommendation is strong. And so I don't know if they're understanding that the amount of evidence that went into those guidelines is as good as it is, or maybe read some of the studies and review articles that informed those guidelines. And so so if your department has any kind of monthly meeting, whether it's grand rounds or any time that the department is sort of getting together and there's a venue at which somebody could speak. My department at Boulder Community had a once a month thing that kind of functioned like that. And so I just asked if it was okay if I shared a little bit of the data and some of the information with the team. And so I shared several studies with them, but for example, Andy Bowdle's group did a dose finding study where you figure out how they gave Sagamedex and aliquots to see how much it took to get to a train of 4 ratio of 90%, which is the standard. And, in that study, they found that 87% of the patients didn't require as much Sagamedex as the manufacturer dosing recommendations would have suggested, but 13% of the patients required more, and almost 0% of the time they got it exactly right. So what I was sharing with my department was like, guys, 87% of the time we're wasting an expensive drug. And there's money that could be saved, or you could just be giving your patient the right dose, which is always great. And 13% of the time we're falling short, and we're taking the patient to PACU with potentially a weak upper airway. Everybody thinks it's the diaphragm, like, oh, okay, they're generating a great tidal volume. That's actually not the problem. The problem is that the airway is weak and narrowed in its diameter. Those muscles are not strong enough for the patient to keep their airway reliably patent and swallow normally. So those patients are at risk for obstructing, desatting, aspiration pneumonia, and at risk for pulmonary complications in general. Not to mention that they will languish in your PACU, and they'll have this story, a slow wake up. Some number of slow wake ups are actually inadequate reversals from neuromuscular blockades. Now, sometimes it's too much midazolam and sometimes it's too much narcotic, but some of those were the patient not getting all the way reversed. And so it was helpful to be able to get in front of the team and kind of explain that. On the economic side of things, if you're talking to one of your administrators or the purchaser, or maybe your CFO, I think they want to know, am I going to get a return on my investment if we go ahead and implement the monitors? And there are actually 3 ways to save money with the monitors. I put it into 3 buckets. And the first one, and we've already alluded to, which is sugammadex savings. So at my hospital, Boulder Community, we invested $30,000 to put monitors in 8 ORs and a couple of NORA anesthetizing locations. And in our first year, we got back $100,000 in sugammadex savings, because these monitors can reduce your sugammadex by 43%. And so that's a return on investment of 233%. So if you're talking to a CFO and you offer him an ROI of 233%, you might get some people's attention. I think usually you're speaking the language, their love language. So you want to put it in those terms. And then at Creighton Bergen Mercy in Omaha, they saved $200,000 of sugammadex spend in 1 year. And at Children's Colorado, they saved over $300,000 of sugammadex spend in 1 year. And in addition to that, if you can reduce the pulmonary complications, if you look up the study that was done at Temple, they determined that of all the surgeries that happened in their institution in a year's time, if you had a 2% rate of pulmonary complications, that added up to about $7 million of extra care that they spent each year. And they determined that they were probably able to reduce that number to something more like 3 to 4 million if they reliably, quantitatively monitored their patients. It's a little bit hard to capture and conceptualize that number sometimes, because it's hard. If you prevented a thing, it didn't happen. So it's a little bit hard to go back and see what didn't happen, but there's reliable evidence there that you could be reducing pulmonary complications. So every PACU reintubation that you prevent, every pneumonia that you prevent, you're saving at least $30,000. And for that same money, you can monitor 1,500 patients. And the last way you can save money is by saving time. So I already mentioned that the PACU stay is shortened by 7%. I looked up some rates of what certain PACUs charge for their recovery room stays. And the first thing about those numbers are all over the map, which was surprising. But I took an average somewhere in the middle, and you could save $120 per case in PACU time if you crunch the math numbers of the 7% shorter PACU stay times whatever they charge for the PACU. And also Dr. Falk and her group that did that study at Children's, where they showed you could save $300,000 in a year. Actually, it's $360 at their facility. They did some math of their study where they added up the cost to implement the machines, and then they also factored in the cost of the disposable that you put on the patient, and determined that you could save $46 per case every time. So if you want to know how much your facility could save, just figure out how many cases you do in a year where the patients get rocuronium, and take it times $46, and that could approximate your savings. And for most patients most places that have more than 3 or 4 ORs, that number is likely to be in the 6-figure range, or potentially well above.
Okay, thank you. That's fascinating. At least it is to me. And I'm sure that we will hear a lot of comments from the audience, and I really, truly look forward to your comments. We're not here to convince or convert anybody, but I really hope in the Q&A session We'll have an open dialogue about what the current problems are, why you perhaps or your colleagues don't believe that quantitative monitoring today is preferred. We asked for some questions from the audience. We got some online, but I'd also like to invite all of you to come up to one of the microphones. Please let us know where you are from, just so we can follow you and appropriately punish you.
Okay. And I'll be devil advocate right now. So my question for panel is, I'm Boris Mraic for the Ohio State University.
May I please say the Ohio State?
The Ohio State. And my question is how the panel will interpret the popular study. The people who are not familiar is the study published in 2019, 28 countries in Europe, over 220 hospitals, 22,000 patients, and they were actually looking at use of muscle relaxant, use of reversal, use of monitoring. So they concluded that baseline is when people don't use at all muscle relaxant. That's the baseline to new pulmonary complications. And then if you use muscle relaxant, increase for odd ratio 1.3, so 30% increase. Then they look, what about muscle relaxant? We reverse them because a lot of people were not even reversed. Didn't change at all. Then they look at it, neostigmine versus sugammadex. That was published 5 years ago, so it was hot topic. No changes whatsoever. Then they actually look at the end, when people did use quantitative monitoring, if it's train of 4 more than 0.9 when extubated, no changes whatsoever. So how would you interpret that? And the pharmacy or CEO of hospital said, here, didn't change. It's real world data, 200,000 patients, 28 countries.
Yeah.
Although I agree with your data, but let's see.
Okay.
What do you answer on that one?
Do you mind if I take this one? And
Yeah, actually, I was going to submit that Dr. Brull, you might be the best one to speak to this one. I'm happy to, but I think that you're going to
The only reason I'm going to respond to it is because I happened to write a letter to the editor about that, which, by the way, was published. And what wasn't apparent in their original presentation, and Manfred Blobner, I've known him for 30 plus years, and all of the authors on that original article. So I tried to be as polite as I could, but I said it was basically rubbish, because out of the 200 and some thousand cases, if you look at it, there were only about 8,000 patients that were monitored quantitatively. All of them were monitored quantitatively with acceleromyography. And if I remember the number correctly, it was, 0% of those patients, they normalized the values. So they may have extubated patients at a train of 4 ratio of 0.9. The problem is that the vast majority of acceleromyography-based monitoring actually starts at 120 and above. And if you don't normalize and you still extubate at 0.9, 0.9 of 130% is not 0.9, okay? So all of the few patients who were monitored with acceleromyography were not actually extubated at the true baseline train of 4 ratio of 0.9. And in fact, in their follow-up popular remassaging of data, they have changed the threshold for extubation from the same patients to 0.95. And at that point, the quantitative monitoring did show an improvement in outcome, pulmonary complications. So that just proved my point. So yeah, and it's fascinating to talk to them.
Can I add one thing, Dr. Burl? Or go ahead, and then I was going to add something to
So
what
just
he said.
comment. So I was in European meeting that published was the study was published just a month before I was presenting pro and con debate using the monitoring, and then my quick interpretation was this study showed the real world data that people don't know what they're doing it at all because it's just terrible.
Yeah,
absolutely.
Yeah,
And especially as panel are presenting, I can tell you much more terrible situation what people are doing with even with monitor that we have in every single OR, and people are just disconnected, don't even use it, still using the manual, and then they press here. Oh, it is twitches, as you said. It's direct stimulation of the muscle. Of course, it twitches.
Yeah, Soren, can you push the number 7 and see if that brings up slide 7? I just want to
see. 7 is that number between 6 and 8.
Yeah. Is that? We were going to try to go to slide 7, or I don't know if you can go forward on the slides until you get to the one with the MRIs of the airway. See if we can pull it up.
5, 6.
One more. Okay. Oh, go back one. These are all good slides to look at, but let's go back one slide. So you brought up a very good point, and this is a point that I think sometimes people might gloss over and not realize it, which is that if any patient is given a neuromuscular blocking drug, they have a higher likelihood of having complications versus patients who were never given a neuromuscular blocking drug at all, like say compared to like you did the case under LMA. So if the patients who were paralyzed at all have more complications than the patients who were not, and that's regardless of your monitoring or reversal strategy. And I want to show you a picture that's worth 1,000 words. So these are a series of images where you can see the width, the caliber of how wide the airway is. So starting with the one on the top left, look at just how wide the black airway is. You can see the black airspace, kind of in front of the cervical spine and spinal cord. There's pink arrows pointing to it. So snapshot that in your mind, how wide that is. And now look at the airways in the next 3 images where you have 4 twitches in all cases, but that airway doesn't get back to anywhere near as wide as it was until you're at a TOF ratio of 100%. And so in cases, there was probably many patients in the study that you mentioned that maybe got partway back to their airway being restored to the way it was before, but not all of the way. So in these middle two pictures number 2 and 3, there are many patients being taken to the PACU with that airway. And again, they can't keep it patent. They can't swallow right. They may feel weak or short of breath. They have double vision. They're confused, and also their chemoreceptor in the carotid body that detects hypoxia is also impaired function until you're back to TOF ratio of 100%. So I would submit that we need to find a reliable strategic method to stop taking patients to the PACU with airways number 2 and 3 on that slide.
So, but do you extubate patient at 90% or 100%?
Well, 90% is the standard, but since I saw these images and I read the information about the chemoreceptors not responding to hypoxia appropriately until closer to TOF ratio of 100, I've become a TOF 100 guy, but I'm still out there trying to get people to at least do 90 or understand why 90 is important.
Yeah. So when I was doing for Sugamarex for Merck, the protocol was three every 15 seconds of 90%. It needs to be 3. One is 89, you need to repeat it.
Yeah.
So I think 3 of 90,
and then
100 could be. Right.
Yeah. The other great thing about this monitor in particular, but just any monitoring is, I think, it's like if you look at your BIS reading or maybe a CVP number, it's better to take 10 or 12 readings than it is to look at just one and be like, okay, I'm good. So I like to, when I see them hit TOF 90, 95, 100, I try to make sure that I've had a couple of readings in a row that are consistent, which is usually not a problem with these much better monitors now. And then this monitor in particular will show you your signal strength in the top left corner. And so when you become a frequent and savvy user of this device, you almost play games with yourself being like, how good can my signal strength be? And the higher that signal strength is, the higher level of confidence you have that you have achieved the goal.
Thank you.
Just a follow-up of what you have discussed. All major cohorts of 1 million or whatever number of patients have shown that patients who have had muscle relaxation have had, increased adverse outcomes compared to patients without it. So definitely reversal is key. But my question is, I'm Pedro Ibarra from Bogota, Colombia. In our hospital, we use devices like TOF guard, okay? And we have the resistance of using it routinely is that every once in a while, we get somebody with, let's say, 38% relaxation at that point, and waiting just a few minutes in order to start with the reversal. The patient might just simply wake up, and sit up. So people start to realize that maybe it's not that reliable. The number is not that reliable, and so might have some resistance to use it routinely. My question is regarding this device, which is EMG technology. How often does these happen? Because, of course, devices are not 100% perfect. How often do you get unreliable readings?
Yeah, so there's a couple of things. First of all, I think it's important to remind people that at train of 4 ratios of 30, 40, 50%, patient can jump out of bed and dance or jig in the corner, but their airways look like this, right? Because those pharyngeal muscles are very sensitive to residual paralysis. They require full strength to coordinate to maintain upper airway patency. And so just because a patient is moving out of bed, just because they're taking good tidal volumes, doesn't mean that they've reached that train of 4 ratio of 90%. 20 years ago, the gold standard was 70%, and patients looked just fine. Patients can look just fine at less than 70%. So that's the first thing. The other thing is, recalibrating a little bit as to when you think patients can move in terms of where they are in the recovery. One thing, using qualitative monitoring, typically you don't start to see diaphragmatic effort until 2 twitches on the train of 4. One thing that we're seeing with this technology is that we have to kind of recalibrate a little bit and get people used to the fact that some diaphragmatic efforts, particularly when you have a laparoscopic surgery and they're looking at the diaphragm, might start to see movement earlier than that at PTC ratios in the double digits. And using that as a guide to redose and saying, oh, well, the PTC says it's 15, the diaphragm shouldn't be moving, but the diaphragm is moving, that means the device is not working. It's probably not the case, right? I think it's just a matter of recalibrating the expectation when you're using quantitative monitoring than what you're used to with qualitative. But I think with your example of TOF ratio is 0.4 and the patient's getting out of bed, I think it's just a matter of understanding the physiology of neuromuscular blockade to say that that's not necessarily showing that the device doesn't work. What it's showing is that there are a lot of muscles in the body that recover more quickly than some of the most susceptible ones, particularly the pharyngeal musculature.
And if I can add one other reason to be at 0.9 or above, it's not just the pharyngeal function, the genioglossus, which is really still paralyzed at 0.9. It's the hypoxic pulmonary response to hypoxia, the hypoventilatory response, is markedly reduced. So you may well have a patient with a train of 4 ratio of 0.8 or 0.9 in the PACU who is hypoxic, and they cannot increase their tidal volume or minute ventilation in response to that hypoxia, because that's a chemical, that's a central process rather than a neuromuscular process. So there's a lot of reasons to consider 0.9 or above as minimum. The fact that patients, jumping off the table at the train of 4 ratio of 0.8 means that perhaps we didn't time correctly the anesthetic, the level of anesthesia, with a level of neuromuscular block recovery. So maybe they need 50 milligrams of propofol to allow them full recovery instead of pulling the tube and running into the potential complications from residual neuromuscular block.
Just
thought.
just because that patient was maybe able to jump out of bed or sit themselves up before they got to train a 490, it doesn't mean that they're going to do well. They actually could still be symptomatic from residual blockade. And so after they get done sitting up out of bed, if you don't finish getting them to train a 4 ratio of 90, they could still be symptomatic in a variety of ways. You know, they can be confused, feel short of breath, have double vision, they can't see well. And so it doesn't mean the patient will do well. Can you go to one slide backwards?
My question regarding is the quality, the distance and reliability of this technology against the other one. Because actually, many of these patients with 30-something, when this happens, you take a new reading, and the reading is now 90%. So within a few minutes, without even any reversal. So my question is regarding the reliability of the figures that you guys with this EMG technology.
Yeah, so there's a lot of different quantitative monitoring technologies you can use, but you know, EMG is one of the best, and I would certainly submit that it is better than the peripheral nerve stimulator from 1970 by a long ways. I had an instance this week where just what Dr. Artime said, where one of my partners texted me that. So this monitor can show you what's underneath of a train of 4 count of 1 out of 4, what's between that and 0 out of 4 twitches. So it can show you the post-tetanic count, which is from 0 to 20. And he said, I have a post-tetanic count of 12 out of 20, which is a deep block. But look at my capnogram. The patient is clearly making a lot of breathing and respiratory efforts. What gives? Well, the couple of things there. So because this monitor will show you what's underneath of the first twitch coming back, or the first twitch is coming back soon, you can start to figure out at what number PTC that diaphragm seems to be waking up. And because you can see more of that information now, you can now refine your neuromuscular block so much better. So I'll find different patients. I'm like, this patient wants to breathe at a PTC of 8 out of 20. This other patient wants to breathe at a PTC of 15 out of 20. That is actually a thing that if you get these monitors, that you will be able to start to see and titrate to that and make the conditions better for your robotic surgeons. And you'll really appreciate being able to see that. So that will erase the age-old robotic surgery conundrum where the robotic surgeon says the diaphragm is moving, and you look at your peripheral nerve stimulator and you say, but there's no twitches. Well, there's stuff under there, and you need to be able to see it. And then once you see it, you can act on it and run your management accordingly. But in the case of my colleague who sent me that, I also explained to him, I'm like, remember, right now you're monitoring the ulnar nerve. The diaphragm can wake up and recover sooner. So if I was able to put sensors on the actual diaphragm, it might be showing a TOF count of 1 out of 4, but I can't do that. So we need to remember which muscle groups are recovering at which speeds. And then lastly, because he sent me a picture of the device, I was able to see that his signal strength was 21.5 millivolts, which is a phenomenal signal strength. If you have that signal strength on EMG, you can take that data to the bank. It's very reliable. And so a lot of times when people look at something they don't understand, they think the device is broken, but I would submit at least half the time something about our understanding is broken. And we need to go back, we need to look in the mirror, we need to go back and remember some certain things about the neuromuscular physiology and what we learned a while back that we maybe have forgotten.
But I'm happy to discuss this and maybe better understand what the question is after this session, if you'd like to stay. Any other comments or questions? Yes, please.
Yeah, I traditionally use the TOF. I don't have much experience with EMG, but I use a TOF card. I'm a religious follower of that. So in fact, I would not reduce my anesthetic depth till I get a 0.9 and then extubate the patient. Few comments, like in the robotic surgery, when we have the diaphragm moving and I see the count as 0, I may choose to use PTC, or I often increase the depth of anesthesia and then just takes care of it. I had a question for the use of TOF in pediatric. Often, I don't always do a single twitch before I use a TOF because to my understanding, TOF doesn't really need determination of supramaximal stimulus. You can even use it with a submaximal stimulus. So even if I don't do a single twitch and determine the submaximal stimulus, supramaximal stimulus, I still can go ahead and use a TOF at the end. You can correct me if I'm wrong. I fail to understand what should be the the amplitude which I should select in pediatric, because there's very little literature with respect to weight of the child and what current strengths you should use, especially if you have not determined a supramaximal stimulus. That was one query I had. Second query, sorry, I have a heap of queries which I've collected. Second query, traditionally in the older guidelines, we said at a twitch of 2, we could go ahead and reverse. That has been now updated to a TOF ratio of 0.4. There's a lot of focus on Sugamadex, and somehow the use of neostigmine has been underplayed. What has been the change or this drift from neostigmine to Sugamadex? Why have we given off the TOF 2 for reversal and gone down to TOF ratio of 0.4? Last question that's just come up with all the discussion which we did, which one of you mentioned that at 0.9, the hypoxic or the response of the carotid body is not completely back. But as you rightly said, that's a chemical response. So how does reversal at the NM blockade affect a chemical response of the body to CO2 is again something which doesn't sink in.
Okay.
Thank you.
I think there's the 3 questions in there.
Many of them were pediatric, so but I don't remember what
Please.
they were.
Do you have a guideline which decides what current
Yes.
twitch strength you use for based on the child's weight? I mean, there's nothing in adults, it's fairly well decided, and you generally start off at 40 milliamperes. But in children, there's no really way to guide unless you're doing a supramaximal stimulus, like how much current strength should I use for a 5 kilo child versus a 10 kilo child versus a 15 kilo child? Like, how do I gauge the current strength that I need to use?
Let the monitor do the work, because the neuromuscular junction is the same in pediatrics as it is in adults. You do need supramaximal stimulation with electromyography because you have to have a starting point to compare against. So there's quite a bit of literature on that, and I'm happy to share it with you if you want to stay afterwards. Any other comments?
No, that's what I wanted to say, that the monitor will.
So because the EMG allows you that, a traditional TOF guard, you have to select the current strength. So when you're using the older monitors, you're at a loss at what current strength I should start off with. That would be one of the limitations of the older monitors.
Right. So you're doing Cal 2, not Cal 1 with the TOF watch. But let's stay and we'll discuss that afterwards. A lot of people may not be interested in this.
To speak to one of your questions, though, you asked about neostigmine falling out of favor. But actually, since I've got this quantitative monitor in my ORs, I identify the patients who recover back to TOF 40, and I pivot those patients to neostigmine, assuming I've got 10 minutes before extubation to let it work. One of the problems with neostigmines is it's much slower because of the way it works. So if you start using this routinely on your patients, what you'll find is that 15, if not 18% of the patients will spontaneously recover their own block, sometimes back to a training forward ratio of 100%, all on their own, and they don't need any drug. About 38% of the patients will get back to TOF 40, and you can pivot them to neostigmine, and you can do so safely. And the other 50% will need Sugamadex, and sometimes more than you think. And there is no way to identify those patients without a quantitative monitor. It is not possible.
And I think it's a great tool to show how neostigmine works. Again, for our trainees, many of them come with only ever having used Sugamadex. And when we have this technology, we can show the difference, show how much longer time you actually need for it to be appropriate to extubate. But it's appropriate to extubate. We're gonna get there. But you have to use it smartly.
Yeah, great teaching tool.
Yeah.
I think it's the time of reversal which is deciding between the two. Last question, sorry. Why do we think that the carotid body will be more responsive at 100% versus 0.9 when that's a reversal at the muscle NM junction and it's got nothing to do with a chemically mediated response?
I think my opinion is when you've made the decision to use a neuromuscular blocker, you have charted a course that patient's going to have, that receptor is going to be abnormal for a period of time. You're right. Even if I get it back to TOF 100, has the chemical sensitivity of that receptor recovered all the way or not? Good question. But for me, I wouldn't want to do anything else other than getting them back to 100 to at least give them a chance to try to restore that function as soon as possible, even if it's not 1 minute after the case ended.
And there's answer to that by Barbara Wad in the 1970s that a 24 recovery, 24 ratio recovery to 0.9, that level that we consider normal function, we're actually functioning on 30% of the receptors that are unoccupied. The other 70% are occupied. It's called the margin of safety. So even at a 24 ratio recovery to 0.9, you're only functioning on 25 to 30% of the original receptors. Barbara Wad.
Thank you.
Absolutely, yeah, absolutely. And that this soup is potentiated, absolutely. Thank you.
Hi, I'm Daniela Damian. I come from Pittsburgh, from Children's Hospital. I have a question regarding how long do you monitor? So some of you said 3, like the measurements at the end to prove that it is above 90% or close to 100. One second, did you ever see weakness in the recovery room after you thinking that you reversed 100% or whatever above 90%, and you gave Sugamadex, let's say, and then did you have cases which you had weakness in the recovery room, and then what do you think the cause for that would be, or even worse, patient getting reparalyzed?
So in the pediatric population, absolutely, I have seen that, and I have to think that it's the way we administer the medication. That oftentimes we use small doses, and some of those small doses in the pediatric population some of it stays in the hub, and the next time somebody flushes that, there could be extra medication going in. So when we use a muscle relaxant or really any drug, I encourage whoever I'm working with, trainees and so on, to make sure you flush it. You don't leave any drugs unattended, basically, in the tubing, because that's exactly what happened to one of our patients. We called to the PACU, patient is struggling to breathe, looks like the patient is paralyzed, but we reversed, we followed the numbers, it was all good, what happened. And when I asked what was flushed lately, and I asked, where did you administer the relaxant originally? And it turned out to be the same port. So I believe there was a little extra stay there, and that caused the paralytic.
Okay. For those of you who still have questions, I'm sorry, we'll take them here. I don't want to keep everybody else who has other places to go, but we're going to be here. So Wade, please come ask the questions.